What's up, TUMNAD lectures? In this video, we are going to be looking at what we call the gastric secretions, and we are going to be mainly focusing on what we call the intestinal phase of gastric secretions. If you find it helpful at the end of it all, please make sure you like the video, comment down in the comment section, and then subscribe. Let's go ahead and get started, okay? In the previous videos, we have been looking at what we call the gastric secretion phases, where we look at what we call the cephalic phase, accounting for the 1/3 of the gastric juice. We look at what we call the gastric phase, which was accounting for the 2/3 of the gastric juice. And now we are looking at what we call the intestinal phase of gastric secretion, which is going to be accounting for a very small percentage of what we call the gastric juice. Okay, it doesn't account for a significant amount of the gastric juice. Okay. So now, during our discussion, we are going to be following these kind of the objectives. So look at what we call the overview of the intestinal phase. We look at what we call the stimuli, and even also look at what we call the inhibitory factors, which is going to be taking a little bit more of the time explaining the inhibitory factors of gastric secretions during what we call the intestinal phase. Okay. First, we need to understand what's going to be the intestinal phase. Now, the intestinal phase is going to be this phase of gastric secretion, which is going to be occurring in what we call the intestines. Okay, it's going to be occurring in what we call the intestine. That is something which is very key. That this phase is happening in what we call the intestines. Okay, when food reaches into what we call the intestines, the intestinal phase of gastric secretion starts immediately. Okay. Now, the point is, we need to first understand how is this food going to be leaving the stomach to go into what we call the intestine for intestinal phase to start. Now, this is going to be our diagrammatic illustration of what we call the stomach, and of course, the stomach is going to be having a different kind of the areas. This area here is what we call the cardia, which is going to be having this kind of the sphincter, which we call the cardiac sphincter. Okay. Then we also have what we call the fundus, okay? Then we also have what we call the body of the stomach, which some other people call the corpus. Then we also have what we call the pyloric antrum, okay? And then finally we have what we call the pyloric canal, okay? Those are the regions of what we call the stomach. Now, how is this chyme going to be moving from what we call the stomach into what we call the duodenum? Now, this one here is going to be now a little of what we call the semi-liquid substance, which we call the chyme. And of course, food, whenever food is leaving the stomach to go into the duodenum, we no longer call it food, but we just call it the chyme. Okay. So now I want to see how this chyme is going to be leaving the stomach into what we call the intestines. Now, what is that kind of the mechanism through which this kind of the chyme is going to be leaving the stomach into the duodenum? We're going to be using what we call peristaltic waves, and of course peristaltic waves are going to be under the influence of what we call the muscularis externa, or the muscle layer of what we call the stomach wall. Now, we're going to be having waves of contractions over the stomach. We're going to be having the fundus contracting, the body contracting, the pyloric antrum contracting, and even also what we call the pylorus canal also contracting. Now, what is unique is that the strongest peristaltic wave of contraction of the stomach is going to be in what we call the pylorus. Okay, the pylorus is going to be exhibiting the strongest peristaltic wave of what we call over the stomach. In other words, this area is going to be experiencing a strongest peristaltic wave, which is going to be squeezing some of the little chyme to bypass this kind of this sphincter. You know how this sphincter is called? And this sphincter here is what we call the pyloric sphincter. It is that gate that allows the chyme, which is ready, which is completely digested, chemically and mechanically, to be squeezed into what we call the first segment of the small intestine, which we call the duodenum. Okay. So now, if we have the fundus contracting, we have the body contracting, we have the pyloric antrum contracting, and then we have a strong contraction from the pylorus, it will squeeze some of the chyme into what we call the first segment of the small intestine. Okay, so now you're going to be having some little of the percentage of the chyme, which is going to be coming into what we call the duodenum. So now you're going to be having this kind of the chyme. It has now crossed into what we call the duodenum. However, the biggest percentage of the chyme is going to be pushed back to what we call the body of the stomach for more chemical and mechanical digestion, until when it is ready to bypass this kind of the canal, which we are calling the pylorus canal, which is going to be gated by what we call the pyloric sphincter. So now, this kind of the contraction is going to be squeezing some of the chyme and going to be pushing back some of the chyme back at what further chemical digestion and mechanical digestion. So now, this mechanism of pushing some of this chyme back, this one is what we call retropulsion. Okay, this is what we call retropulsion. Okay, that back movement of the chyme for proper chemical and mechanical digestion in what we call the body of the stomach. Now, we have now seen how this kind of the chyme has moved to what we call the duodenum. Now, the question is, we need to first go ahead and see what are some of those components, what are some of those molecules that are going to be within this kind of this chyme. It is moving from the stomach into the duodenum. Let's go ahead and first see some of those kind of the molecules that this kind of the chyme is going to be coming with. Remember, this area is super concentrated with what we call hydrochloric acid. So now this chyme is going to be moving with what we call the acids, and this one is what we call is going to be having what we call protons. Okay, it's going to be having what we call high concentration of what we call the protons. Okay. At the same time, this kind of the chyme is going to be containing partially digested proteins. And the beautiful term we can use here is what we call the peptones. Okay, so now this chyme is going to be having what we call the peptones. These are partially digested proteins. And of course, don't forget, it's going to be having what we call the fatty acids or the fats generally. Okay, it's going to be having what we call the fatty acids. Super very important. And then the last one is going to be having short chains of what we call the carbohydrates, which we may call the oligosaccharides. Okay, so we're also going to be having what we call oligosaccharides. Okay, Okay, so now these are the key important molecules that is going to be contained in what we call the chyme that has come into what we call the duodenum. Now, within the mucosa of the duodenum, we're going to be having a couple of different cells, and these cells are going to be super sensitive in these different kind of the chemicals or this kind of the molecules that has come along with what we call the chyme. And it will stimulate different cells, which will lead into the gastric secretions, and some of which will lead into what we call inhibition of the gastric secretions. So now, let's first go ahead and see the stimuli. What is these molecules? Which cells are they going to stimulate, which will lead into what we call an increase in the gastric secretion? But of course, at the back of your mind, you have to know that it's going to be a temporary release of what we call gastric secretion. It's going to be very small percentage of the gastric juice. Okay. Now, within the mucosa of this duodenum, we have this kind of this cell. This cell is super very important. And this cell is what we call intestinal G cell. Okay, what we call intestinal G cell. This cell is super sensitive to what we call high levels of what we call partially digested proteins, which we call the peptones. Okay, so now when peptones come in contact with this cell, we are calling the intestinal G cell. Guess what's going to be happening? It's going to be releasing a molecule called gastrin. Okay, it's going to be leading to the production of a hormone which we call gastrin. Now, the point is, we have intestinal G cell. We also have some other cell which we talked about during the gastric phase, which was staying in what we call the pyloric antrum. This one here is also a G cell, but this one is specifically what we call the antro G cell. Okay. Now you may ask yourself, what's going to be the difference between these two cells? Now, the difference is going to be generally in what we call the chemical composition of what we call the amino acids. This one is going to be containing approximately 17 amino acids. So now for this case, this one is going to be what we call G17. But for the case of the intestinal G cell, is going to be having a long chain of amino acids. Generally, it's going to be what we call G34. That's going to be the difference, but the effect is going to be the same. Okay. Now, what happens that these intestinal G cell, when they are stimulated by peptone, they are releasing what we call gastrin. Gastrin is a hormone. It's going to be flowing to the blood. It moves through the blood. It goes to these kind of B cells. These cells are super very important. You know how these cells are called? This cell here, this one here is what we call the parietal. Cell. And then this cell down here is what we call the chief cell. These cells are super sensitive to what we call gastrin. what happens is that a gastrin has a specific receptor on this cell we are calling the parietal cell. It will stimulate the parietal cell through a signaling mechanism which will lead into an increase in intracellular calcium, which will stimulate this proton pump, allowing the exit of what we call hydrochloric acid. Okay, so now this one is going to be our proton pump. When this proton pump is stimulated, it will release what we call the protons into what we call the stomach lumen, which accounts for what we call the gastric, what we call the hydrochloric acid. Okay, such a beautiful thing. Now, what happens is that the gastrin can also move into the blood and go to this the cell we are calling the chief cell, and then it binds on the specific receptor on the G cell, which will stimulate a signaling mechanism, which will lead into an increase in intracellular calcium. When there is an increase in intracellular calcium, within this chief cell, we have this vesicle, which is going to be containing one of the proteolytic enzyme, which is in active form. You know how this proteolytic enzyme, how it is called? This one here is going to be a vesicle containing what we call pepsinogen. Now, under the influence of increased intracellular calcium, this vesicle containing pepsinogen is going to be fusing with the apical membrane of this cell, which will lead into the exocytosis of this molecule we are calling pepsinogen. Okay, so now you're going to be now having a release of this molecule we are calling pepsinogen. And of course, we have said that pepsinogen is an inactive proteolytic enzyme. How is it going to be getting activated? Now, pepsinogen, under the influence of the pH or the acid, pH between 1.5 to around 3.5, this pepsinogen is going to be activated into another active molecule, and this molecule is what we call pepsin. Okay, is what we call pepsin. So now we have said that this influence is going to be under the influence of the pH of between 1.5 to around 3 to around 3.5. So now within this pH, we convert pepsinogen into an active pepsin, which starts the chemical digestion of what we call the proteins. Okay, such a beautiful thing. Now, what happens is that if the pH goes on increasing and increasing and increasing, it is going to be converting pepsin back into pepsinogen. Okay, now if the pH goes extremely too high, for example, up to around 7.3, okay, it's becoming too alkaline, and this one is going to be more of an irreversible reaction. What happens is that pepsin is going to get converted back into pepsinogen, and it will never get converted back into pepsin, even when the pH goes back between 1.5 to 3.5. Okay, such a beautiful thing. Okay. So now that's generally how gastrin is going to be causing that the influence, leading to what we call an increase in the gastric secretion. But at the back of your mind, you have to know that this is going to be a very small amount of the gastric juice secreted by this influence. Okay. Then we also have another this cell. This cell, they don't know even its name up to now, but this cell is super sensitive also to what we call high levels of what we call peptones. Okay. When peptones come on this this endocrine cell, it releases one of these molecules. You know how this molecule is called? This molecule, which is going to be released by this cell, is what we call enteroxythin. Okay, what we call enteroxythin. Now, enteroxythin, this molecule also flow into the blood. It will move and move and move and move. It has specific receptors on this cell we are calling the parietal cell. And when this molecule called enteroxythin binds on this receptor, it will stimulate what we call the parietal cell by signaling mechanism, which will lead into an increase in intracellular calcium, leading to the activation of this the proton pump, leading to the secretion of the hydrochloric acid. Okay, such another beautiful thing. So now that's generally how the stimulatory factors of what we call gastric secretion during the intestinal phase. Okay, now the proceeding mechanisms are going to be more and more of inhibitory factors. You're going to be taking more little with the time and understand the inhibitory factors of gastric secretion during the intestinal phase. Okay, because this intestinal phase, it is trying to inhibit gastric motility, gastric secretion, to make sure that these the intestines are given enough medium that helps in what we call the chemical and then the mechanical digestion, because the enzymes which we shall talk about, which needs to come and run the process of chemical digestion in the small intestine, doesn't need what we call the acidic medium. We need alkaline medium to favor their kind of the activity. So now we shall limit the exit of what we call the chyme from the stomach into the duodenum. That's the significance of what we call the intestinal phase of gastric secretion. Okay. So now let's go ahead and see the inhibitory factors of what we call the intestinal phase of gastric secretion. Now, within the mucosa of this kind of the intestine, we have this kind of this cell. This cell is super very important. You know how this cell is called? This cell here is what we call the S cell. This S cell is sensitive to a lot of different molecules within this chyme. You know, with this kind of these molecules, this cell is sensitive to what we call high levels of the protons. That is one. It is also sensitive to what we call the fatty acids. Whenever these two molecules come in contact with this cell becomes activated. And when it becomes activated, it will release a hormone. You know how this hormone is called? This hormone is what we call secretin. Okay, it will release a hormone which we are calling secretin. Okay, what we are calling secretin. Secretin is a hormone which is going to be now flowing into the blood. Now, when it flows into the blood, where is it going? It is going to come over here and then it comes on this kind we are calling it the antro-G cell. It has specific receptors on this kind of this antro-G cell. It will come over on this kind of the antro-G cell and inhibit this antro-G cell from producing what we call the gastrin. And if you reduce gastrin, that means you're going to be reducing the stimulation of the parietal cell and the stimulation of what we call the chief cell from producing the kind of the gastric contents. Okay, so now in that way we are seeing that when you inhibit the antro-G cell by the influence of secretin, you are reducing gastric secretions. Okay. Besides that, this secretin can also move direct and come over on this kind of the cell we are calling the parietal cell and inhibit the parietal cell from producing the hydrochloric acid. It can also come over on this chief cell and also inhibit the chief cell from producing hydrochloric acid. Okay, such another beautiful thing. Okay. Now, this secretin, it has also another kind of the mechanism it does. You know what that one does? This kind of secretin, because it is secreted following sensitive to what we call fatty acids, it means we need some kind of the molecules that are going to be breaking the fat globules into tiny molecules that can be acted on by what we call the pancreatic lipase. You know how that molecule is called? That molecule is what we call the bile. What happens is that secretin will move over into the blood. It will move and move and move. It comes this kind of this structure here. This structure we are calling the liver. It has specific receptors within what we call the liver. What happens is that secretin is going to be running the process of synthesis of what we call the bile acids, mainly from what we call the cholesterol. If you remember very well, when we are talking about bile acid synthesis, we said that cholesterol is going to get converted into primary bile acids, the chenodeoxycholic acid, the cholic acid, which will eventually lead into the formation of what we call the bile salts. Okay, so now the bile salts are the ones which are going to be responsible in what we call emulsification of fats. Now, if you have secretin coming to bind on these specific receptors on what we call the liver, it will increase the synthesis of what we call the bile. Now, the bile from the right liver lobe and then the left liver lobe will pass through what we call the right and then the left hepatic duct, which will come together to what we call the common hepatic duct. Of course, the common hepatic duct will unite with this kind of the duct we are calling the cystic duct into this kind of this structure which we call the gallbladder. And then from there, it will move down to what we call the common bile duct, and then eventually comes over into this kind of this structure. You know how this structure is called? This structure here is what we call the pancreas. We're going to be having this main pancreatic duct and this kind of this bulbous structure. We shall see them later. Okay. Now, what happens is that our bile is going to be moving down and down to the duodenum. And when it enters into the duodenum, it's going to start running the process of what we call emulsification of fats. Okay. Such a beautiful thing. Now, what else? That secretin also has another kind of the mechanism. It moves into the blood and it goes and comes on over specific type of cells within what we call the pancreas. So now, what happens is that we are going to be having what we call the ductal epithelial cells. Okay. Now you're going to be having these ductal epithelial cells. They're going to be having receptors of the molecule you're calling secretin. What happens is that the secretin will stimulate these receptors on these ductal epithelial cells, leading to the production of what we call the bicarbonate. And of course, bicarbonate will flow what we call the main pancreatic duct, and then to come over on this kind of the duodenum and neutralize all the protons. This is how it's going to be happening. Now, you're going to be having now the bicarbonate. You're going to be having the bicarbonate coming from the pancreas. It will react with what we call the protons, which are coming with the chyme. leading to the formation of what we call carbon dioxide, and then also what we call water. Okay, so now the carbon dioxide can be moved out through the breathing mechanism by the lungs, and then also the water is going to be running other kind of the mechanisms within what we call the intestines. Super very very important. Okay, so that's generally for the case of the secreting. Now on the mucosal lining of the duodenum, we have this other kind of the cell. This cell is super very important. This cell is what we call the eye cell. This cell is what we call the eye cell. This cell is super sensitive to the following molecules in chyme. It is sensitive to what we call high levels of what we call peptones. Okay, it's sensitive to high levels of the peptones. It is also sensitive to high levels of what we call fatty acids. Okay, it is also sensitive to what we call high levels of fatty acids, and even also it is sensitive to what we call short chains of the carbohydrates, which we may call oligosaccharides. Okay, which we may call oligosaccharides. What happens is that these cells, they're going to be activated by these kind of the molecules in chyme. It will lead into the release of a molecule which we call cholecystokinin. Okay, to release into a chemical which we call cholecystokinin. Now, cholecystokinin is a hormone. It will flow to the blood. It will come and bind the specific receptors within the parietal cell and inhibit the parietal cell from producing hydrochloric acid. Okay, so now again increasing an inhibitory effect of what we call parietal cell from producing hydrochloric acid. Such a beautiful thing. Okay. Now, what happened is that again, this CCK can also move into the blood and move and come over on this structure we are calling the liver. Now, on the liver, this kind of the CCK is also going to be having receptors within what you call the liver. What happens is that CCK will bind on these receptors on the liver and potentiate or enhance the synthesis of the bile acids. Okay, it will also enhance the activities of what we call secretin, increasing more of the synthesis of what we call the bile acids bile salts. Okay. Now, what happens, the bile acids, bile salts are going to be moving through what we call the biliary system. It will come over to the duodenum and start the process of emulsification of what we call the fats. Okay, such beautiful thing. Now, what else does CCK do? This cholecystokinin has receptors on this structure you're calling the gallbladder. It will bind on specific muscle layer of the gallbladder and squeeze it to produce out what we call the bile, which will move to the common bile duct down to what we call the duodenum for the process of what we call emulsification. So now CCK can come over and bind on the receptors which are going to be located on what we call the muscle layer of what we call the gallbladder. Okay. Now, what else does it do? Now, we also have CCK coming over the pancreas to come on this kind of this bulbar structure. You know how these bulbar structure are called? These bulbar structure are what we call the pancreatic acinar cells. Okay, pancreatic acinar cell. Now, I'm going to be cutting a very small portion of the pancreatic acinar cell. We zoom it out and see the kind of the substances that are going to be contained in this kind of this pancreatic acinar cell. So now, this is one pancreatic acinar cell. It's going to be containing a lot of vesicles, which vesicles are going to be containing a lot of different kind of the enzymes. Okay, so now what are some of these enzymes that are going to contain these vesicles within the acinar cell? You're going to be having the following. We're going to be having what we call the pancreatic lipase. Okay, pancreatic lipase. This one is dealing with the final chemical digestion of the fats. We're also going to be having what we call the pancreatic amylase. Okay, pancreatic amylase. This is going to be breaking what we call the final chemical digestion of the carbohydrates. We're also going to be having what we call the pancreatic nucleases. Okay, pancreatic nucleases. This is dealing with the chemical digestion of the nucleic acids. And then the last one is what we call the pancreatic proteases. Okay, pancreatic proteases. This one we are going to be having a lot of them. They are aiming at what we call final chemical digestion of what we call the proteins. Okay. So now CCK is super very important. It has specific receptors on this pancreatic acinar cell, leading to the production of this kind of the enzyme. Those enzymes will move through what we call the main pancreatic duct, and then they come over to the duodenum and they start the chemical digestion of those different nutrients which I've just talked about. Okay, such a beautiful thing. Now, another thing is that we also have another kind of this is super very important cell. You know how this cell is called? This cell is what we call the K cell. Okay. Now this K cell is super sensitive to the following molecules within what we call the chyme. It is sensitive to high levels. It is sensitive to high levels of what we call oligosaccharides. Okay, sensitive to high levels of oligosaccharides, and even also it is sensitive to high levels of fatty, what we call fatty acids. Okay, what happens that these cells, whenever they come in contact with these levels of oligosaccharides and fatty acids, it will stimulate these K cells. Guess what? Which molecule is going to be produced? It will lead to the production of a molecule which we call glucose-dependent insulinotropic peptide, which some other people call the gastric inhibitory peptide. The beautiful name is glucose-dependent insulinotropic peptide. This molecule it can move into the blood, it comes over on this cell and binds on the specific receptors on the parietal cell and inhibit the parietal cell from producing hydrochloric acid. Okay, such a beautiful thing. Now, what else does gastric inhibitory peptide do? Gastric inhibitory peptide also can move into the blood and comes over on this structure according to the pancreas on these small pieces which accounts for 1% of the tissue that make up the pancreas. What happens is that this tissue is what we call the islets of the Langerhans. It has specific type of cells. The cell of the interest in this case is what we call the beta cell. Now, these beta cells, they're going to be having receptors of what we call gastric inhibitory peptide. It will stimulate these beta cells to produce a hormone, and this hormone is what we call insulin. Out of which insulin is going to be one of the most important hormones that helps in what we call reduction of what we call the glucose levels. Now, what happens is that insulin is going to be running a lot of different physiological processes. For example, it's going to be dealing with what we call glucose uptake, okay, what we call protein synthesis, glycogenesis, lipogenesis. So now, insulin is going to be doing a lot of what we call different processes being stimulated by gastric inhibitory peptide on what we call the beta cells. Okay. To sum it all, we also have some other kind of the cells. I'm not going to be taking much of the time, but these cells are also going to be super very important. These cells are what we have this cell which we call the Elo cell, and then also this kind of the cell which we call the Any cell. Now, these cells, they are sensitive whenever there's too much of the chemical that come to what we call the duodenum, which will lead to that kind of little the distension, that kind of the stretch. These cells, they become activated and they release different kind of the chemicals. And the Elo cell is going to be producing a molecule called peptide Y, while the Any cell is going to be producing a molecule which we call neurotensin. Okay. These molecules are also going to be flowing to the blood. They will come over and bind on specific receptors on the parietal cell and inhibit the parietal cell from producing hydrochloric acid. Okay, so now that's why we said that the significance of this intestinal phase of gastric secretion is doing more of inhibitory effect to the production of what we call the gastric juice. We want to give enough time to the intestines for final chemical digestion by giving it a conducive atmosphere to complete the chemical digestion process. Okay. Such another beautiful thing. So now, TUMNAD, we have talked a lot of things. And if you think whatever I've talked is making sense, you're benefiting, please, you can support me. And that one can be done by a couple of means. You hit the like button, comment down in the comment section, and then subscribe. TUMNAD, as always, until we meet next time.